value might change once we get our own measured values from our system. So going back to this conceptual diagram, thinking about our mussels here, we can say that they're increasing in availability in the system, and in some sites decreasing phosphorus availability, but only that was at 4 out of the 9 sites. Now, one thing you have to realize is that mussels aren't distributed across the whole river. They don't have a homogeneous distribution. They're very heterogeneous, where they only occur here, here, here. They're not everywhere. And so this is really important for systems because heterogeneity is really important for maintaining species diversity and just making these systems more interesting. And so to look at this spatial heterogeneity, I digitized the entire Kiamichi River, the main stem, from Big Cedar to Antlers, and GIS, so pretty much traced the entire river using aerial photography. And then I mapped the extent of 46 mussel beds that I knew about within this stream system. And so I had GPS points of all these sites and the entire area that they covered. And so I estimated the aerial excretion, so how much they're peeing, and the aerial storage, so how much nutrients they're storing. And then I also used this metric called volumetric excretion to quantify temporal variability, so changes over time. In volumetric excretion, it estimates the mean addition of nutrients across a given stream reach, so it uses the aerial excretion rate, the reach area, so the length times the width, and then the travel time through the reach. So if you dropped an orange in the river, how long would it take to go 100 meters? And then divide that by the volume of water in the system. And so this gives you an idea of like the entire impact of an organism on that volume of water. And so this is kind of what the digitizing looks like. The mussel beds you can see here in red. And so just moving into the results. So we found that across the system, mussels are storing a lot of nutrients. So this is moving from upstream to downstream. And this is storage of nitrogen, and this is storage of phosphorus. The dark bars are the shell, the gray are the tissue. And so across the Kiamichi, across this river, these organisms are storing approximately 4.7 metric tons of nitrogen. So that's a lot of nitrogen that they're just keeping in the system. And approximately